Hey guys, so welcome to the channel. Today I am just going to do a basic overview of plant breeding. It's my profession. It's what I have a lot of experience in. I have a lot of other interests, but it's probably one of my main ones. I've gotten a lot of awards in it, and I figured since I'm also a gardener, and hopefully you are too, that I can do this basic presentation and show you how you can use this knowledge to help in your general knowledge of plants, but specifically if you're a gardener. So without further ado, let's get started. Uh, Plant breeding, it's really a very ancient profession. It was started uh, thousands of years ago before we even had a word for it, before it was even called plant breeding. In fact, some of the earliest, you could argue, uh, plant breeding efforts were early hunter-gatherer humans who simply found the best yielding or best performing crops or whatever had favorable traits, and they would harvest those crops, and wherever those early hunter-gatherer humans were living, occasionally a seed or two would fall or was deliberately planted. And over time, you would see a slight improvement of that crop to favorable conditions that uh, humans wanted. And that gets to our first question of, what really is plant breeding? And it's basically, we're basically modifying plants to have some type of benefit to humans. Think of it like a video game mod or something, or uh, giving some kind of mod to your car or something like that. It's basically taking some plant in its natural evolutionary state in the wild and really exploiting it for human purposes. We have the official definition here: the intentional manipulation of plant species to create desired genotypes and phenotypes for human benefit. And you might already be wondering, well, what's a genotype and what's a phenotype? Uh, in this channel, I really want to make sure that I explain things clearly and simply without dumbing things down and without using a lot of jargon. One of the best ways to do that is via analogies. So let's actually start with phenotype. So if you're wondering what a phenotype is, that's what you see. So let me give you an example. Let's say we have um, let's say we have a person, and this person has brown eyes. But one of the person's parents has blue eyes, which is a recessive trait. Uh, that person's phenotype is brown eyes, because that's what you see. That's what has gotten expressed. Now, that person's genotype, and this gets into an explanation of what that is. That person's genotype is actually brown blue. It's essentially what is actually there, regardless of it's expressed or not. It's just what is in actual genetic code, and also really what is the genetic potential. So, uh, what does that mean? What is genetic potential, right? Well, if that person who has brown eyes but had a blue-eyed parent, if they had children with another person who had a blue-eyed phenotype, then the resulting children would be they would have some brown eyes and some blue eyes. And so when you have a recessive trait, uh, you need those traits to be homozygous to be shown through. And maybe I'm jumping ahead of myself here, but um, we'll get into that later. But essentially, remember, phenotype is what you see. Genotype is what is actually there behind the scenes. Now, so much of plant breeding is done through controlled pollinations or artificial pollinations. And when we talk about the history of plant breeding and the nomadic ancient humans transitioning into settled agriculture, A lot of this happened in the Fertile Crescent. Populations of people weren't moving around anymore, and so they couldn't go out and find if, for example, if in the local area they weren't satisfied with the quality of uh, the dates from palms that they had, they would no longer just go and search for miles and miles until they found a palm tree that produced good quality dates. They would in fact actually make the palms that made really excellent quality dates. They would actually make those varieties and grow them locally so they would no longer have to travel. And there's a lot of benefits to this. Traveling, seeking out, and foraging, it requires a lot of calories. And especially in ancient times, caloric content is really something valuable. Um, it was nothing like the modern times where many people, not all, but many people do have easy access to at least basic food and calories. In these ancient times, you really had to be smart with uh, every little bit of time and energy you had. And so, um, while they were settling uh, their local communities and becoming localized and no longer nomadic, we don't know how exactly they got started, but they either started with what they had in the local area or on their journey. This is just a hypothesis of mine. I'm assuming they probably would go out on expeditions or on the way to their eventual settling spot. They picked these ideal versions of crops, and as I mentioned earlier, either planted the seeds or the seeds just happened to fall around and grow somewhere inadvertently. But eventually, you'll see as early humans were no longer nomadic, that agriculture really does start to become organized. And you see this around ancient Assyria and Babylon, and um, that's about 1000 BCE. And to put that in perspective, that's about 3000 years ago. So you could say we've been doing controlled agriculture, controlled plant breeding and pollination for almost 3000 years now. That's quite a long time. However, it wasn't until recently that a famous monk named Gregor Mendel started to notice some really interesting patterns in peas. He noticed that they had different traits. Some of them were tall, some of them were uh, had different colors. They had different types of wrinkles in their actual the peas themselves. And he noticed that if he would save the seeds from a pea that was tall and had white flowers, that most of those seeds that would result from that plant would be just like the parent. They would be tall and have white flowers. Now, he started doing experiments where he had other, there's many types of traits he noticed in peas, as I mentioned. So he would take something like a dwarf or a short yellow flowered pea, and he would cross that with a tall white flowered pea plant just to see what would happen, just as an experiment. And he noticed that, the first generation, they tended to favor one parent. And you'll see this in biology. A lot of traits, which come from genes, we'll, get to that in a second. But a lot of traits, not all, especially when we're talking about Mendelian genetics, are dominant. So that means you either have it or you don't. Now, there's other types of traits. There's uh, overdominance. There's incomplete dominance. Where you would uh, cross a red and a white flower and you would get a pink flower, for example. But in reality, genetics is usually not that simple. But in this case, with Gregor Mendel and his peas, most of the traits were, as we say, Mendelian, named after him. They had very simple inheritance patterns. Now, at first, in that first generation, as I mentioned, they look like the one of the parents. The tallness is a dominant trait in biology usually, and believe it or not, contrary to some textbooks, white is not always a recessive trait. I believe in peas, it is recessive, but a lot of my background is in rose breeding, and in roses, it's actually dominant, believe it or not. But in this case, he found something interesting. So all the plants were tall, like one parent, and they had the yellow flowers of the other parent. And so, well, he wondered what would happen if, he continued the experiment. So the next generation, he would save seeds from this hybrid plant between the two different types of peas. And from there, he noticed that you would get all kinds of combinations. And that's why they say the magic happens in the F2 generation and F3 in plant breeding, because you really get to see a great segregation. So in the second generation, you see everything really uh, shake itself up like a Powerball or Lotto, and you can get any kind of combination. So in that second generation, he got plants that were, like the original, they were tall with white flowers. He got some that were dwarf with yellow flowers, like the other parent. But he also got some that were, like the first generation, the tall with yellow flowers. But he also got some that were dwarf with white flowers. And he also got some plants that were tall with yellow flowers. So all basic, combinations for those two traits he would find. And then he began to realize that there must be something going on here. There must be some inheritable unit that is going on that's controlling this. And this is really the first discovery of what we would call in modern times genes. And genes are basically you think of it like a page in a book. You as a person, your entire DNA, or any living biological organism has, for example, let's just say a warehouse full of filing cabinets. Now, your entire DNA set, that's the warehouse. And your chromosomes, these are like the sections, the groups of DNA that are physically together. Let's just call those like filing cabinets. And as you get further and further focused within a filing cabinet, well, you have different uh, books within those filing cabinets. Now, some books are large, some books are small, maybe one or two pages. But each individual gene can be thought of as like a page. And it may not make a lot of sense, but when any mating or breeding occurs in biology, uh, as we know, you get 50% of your DNA from one parent and 50% from the other parent. So it's like taking the entire warehouse and randomly selecting 50% of the pages from all of the books and then donating them to an entirely new warehouse. And there's some other warehouse that donated randomly 50% of its pages. And so now we have this new warehouse that is compiled of books that may be similar. They have portions of either warehouses files, but it's something entirely new. And uh, as you can imagine, because the books might be 50-50, 20%, 75. They're still in predictable chunks, which is why you get things on the first generation so similar looking to the parents. For example, if there was a book for Shakespeare's Romeo and Juliet and one parent, and then the other warehouse donated 50% of a cookbook, in the first generation, you would get a book. It wouldn't make a lot of sense to us, but the book would clearly be half Shakespeare and half a cookbook. Now, in plants, it's a little different because plants are able to self-pollinate. They essentially are able to breed themselves. Most of them, not all, or maybe not even most, but it's a common thing in the plant kingdom. And so that's essentially like taking our warehouse and just copy and paste it. So now we have two of the exact same warehouse, and we're going to randomly select 50% of random pages from each of these books in this new warehouse. again, to make a second generation of book warehouses. Now, in the second generation, you're really going to get to see a true mixing. It no longer will be as easily distinguishable as, okay, this is like half Shakespeare and half cookbook. You might have a recipe, for example, that is written in the Shakespearean style, or you might have a Shakespeare layout type of book, but is written in very dry, dull recipe format. And that's why they say the magic really happens in the second generation plant breeding. It's when the true mixing really happens. It's not always so much in that first generation, but it's a really interesting part of plant breeding that will really allow us to manipulate and create certain new crops and with have traits that we desire. Um, so speaking of traits, I mentioned genotype and a phenotype, what's observable and what's actually hidden. Now, even within those traits, uh, whether they're simply inherited or it's a complex trait, like a measurable spectrum, like height, flower color in peas, for example, are simply inherited. We have qualitative traits and quantitative. So when you hear quant, think of a spectrum, quantitative. When you hear qualitative, just think of you either have it or you don't. So the proper, I guess, visualization or mechanism that I always like to explain this by is by saying qualitative traits are like light switches, on and off. They either have it or you don't. And quantitative traits are really like the light dimmer. It's not a matter if you have the trait. It's really um how much do you have, if that makes sense. And so in Gregor Mendel's case, this is where it can get a little complicated. In Gregor Mendel's case, his P's they inherited height, but height was qualitative for him. So it wasn't always easy um to determine which is which, but in his case, it was simply inherited. But other plants and even animals, height is a quantitative trait. Specifically in humans, height is a quantitative trait. Now, when we get more into the actual active uh, uh, art and science of plant breeding, the important thing to keep in mind is, well, okay, what is your goal? Um, Now, you don't have to have a goal, especially if you're a backyard gardener. Um, some of the most fun I've had as a plant breeder is when I just crossed two of my favorite plants with not having any particular goal in mind. I just wanted to see, what would happen. And that's okay. Now, I've also been in the professional environment, as I mentioned, where you do have to have a goal, and in many cases, it's multiple goals. And so you're choosing parents based off of your goals. So, and that's selection. Selection is choosing um, it's choosing your parents that you want to have as a founder, the founders of your new population, your new uh generation of crops that you're making. So, for example, if I'm trying to create a tomato that tastes good but is also disease resistant, well, then you would pick the as one parent your tomato that tastes really good, uh, and the other parent that would maybe be disease resistant but doesn't have the best flavor, and 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 vice versa. Now, the actual act of crossing these two parents is called hybridization. And a little bit of a footnote here, true hybridization in the strict, botanical and plant breeding science meaning of the word, it's actually a cross between two different species. So when we're crossing, for example, a tomato with a tomato, it's actually just called crossing. It's not a true hybridization. A true hybrid is something like a a mule, which is a cross between a horse and a donkey, or there's a plant called miscane. It's a ornamental landscape grass called miscanthus that you can cross with sugarcane. They're two entirely separate species, and that hybrid is itself a new species. That's what makes a hybrid. It's a combination of two species. It's like a liger. You've all seen um, a liger, right? They're a cross between a lion and a tiger. And if you notice here, there's something really interesting about hybridization and even crossing that happens too. Uh, it's called hybrid vigor. Uh, officially the term would be called heterosis, but if oftentimes you'll have two different species or two different unrelated varieties, that first generation hybrid, that F1, it's going to be really vigorous and very healthy, and it's called hybrid vigor. It's the same reason why you'll see many purebred cats and dogs, for example. Uh, they may be purebred in certain traits, but they really have a lot of genetic diseases and will have a lot of health problems. And you're not going to see these or as much of these problems in a mutt dog, for example. And so it's always a balancing game. It's how do you keep that vigor, but also keeping traits pure, and that's how we get into the art of plant breeding. It's people think it's just a science, but it's not just a science. It's very much an art. And so one way that we do that is by back crossing. So think about the tomato I mentioned earlier. One of the tomato parents doesn't taste good, but it's very disease resistant. And now the other tomato parent is not disease resistant at all. Its leaves fall off all the time, but it manages to make a tomato and it's an heirloom like a brandy wine or your Cherokee purple, and it tastes really good. Now that F1 hybrid, it's not it. I can almost guarantee you, it's not going to be the best of both worlds. So you might be tempted to think that, okay, well, our first generation tomato, it's going to be completely disease resistant, and it's going to taste really well, taste really good. Sorry. Um, And that's not the case. And the reason is because, like I mentioned, yes, some of these traits are Mendelian and simple, but a lot of them are complex, especially when you're talking about something like disease resistance and flavor, which are very, very quantitative and involve sometimes thousands of genes. So that first generation F1, filial, by the way, F is Latin, filial, it means like son. That first generation cross, you're going to get, it's actually going to be, think about this, it's 50-50. So it's going to have some flavor, not as good as other parent, and it's going to have some disease resistance, but not as good as disease resistance as its other parent. So it's going to be more balanced out. Now, you could self it, let it self itself, like Gregor Mendel did, and see what happens in that F2 generation, and you would get plants that uh, would be revert to like one of the grandparents and have new combinations. They might uh, tastes 75% good and have the 25% improve disease resistance. uh but in something like this, if our goal is to create a plant that has both, remember that we want to uh really want to like change our strategy and focus on our goal. So let's say our donor plant is the disease resistant tomato, and the target plant is really our flavorful tomato, and we really just want to get some more disease resistance in there. So instead of taking that F1 and letting it self-pollinate, we're going to back cross it to our original parent that tastes good. And so this drastically changes things. So now in this next generation, you're going to start to see a lot of that flavor return. But you have to be careful because you got to make sure that you're not losing that improved disease resistance. Now, it can get really complicated, like I said. Like, how do you balance it out? If you go back and try to get that flavor, are you gonna like further dilute your disease resistance? Well, the answer is you never know unless you try. Now, these days, there have been so many academic studies and and research done that we can typically tell uh, which disease resistance traits um, are qualitative or quantitative. Um, for example, in corn or wheat, a lot of the disease resistance genes come down to one simple gene. It can be easily inherited. Um, and in tomatoes, that is generally true too. However, there are other cases where, for example, a you have a viral resistance or some other type of resistance where it is like a complex trait. But in this case, we're assuming that the disease resistance we want, that it's a qualitative trait. It's a light switch. You either have it or you don't. So in this second generation, the 75% generation, we're assuming, or we're going to grow out all of the offspring, and we're going to assume that most of them have pretty good flavor. We're going to taste test them anyway, but we're gonna really just going to assume that most of that flavor has been returned. But we'll be able to know which of the this generation, which of the plants inherited that disease resistance from the grandparent, because the leaves will just fall off, they'll just die. And so what you would do is, as a plant breeder, or even as a backyard breeder, which is what I want this geared towards, is you just grow out all the plants and you don't taste test anything until you see which ones have the disease resistance. So by doing that, you're only going to be taste testing plants that we know have some form of improved disease resistance. And we can keep going and going and going with this. So the degree of improvement will decrease every time. So, uh, you might get a smaller improvement if you take that 75% flavor generation and back cross to the really flavorful tomato. But eventually you'll get to something that, for example, UC Davis has done with grape breeding. They have uh, created European style grapes like Pinot Noir and Cabernet Sauvignon, but have have like 97 to 99% of those traits, but they've incorporated those that 1 to 3% of the some wild grape species or some other native grape species that has resistance to, and in this case, I believe it's Phylloxera is the insect that travels Pierce disease. So they were breeding for Pierce disease resistance. So eventually, you would start to get something stable once you keep crossing, and this comes the fun part is, once you get that stability, you have an entirely new variety that you can grow yourself and share with family or friends, and then you don't have to worry about the plant breeding anymore. You can just keep growing your seeds from those crops every year, and uh, your efforts will have paid off. And that, in my experience, really makes it worth it. Now, if you wanted to, we can go very deep into plant breeding. I just wanted to, briefly touch on this. What I've described so far is really basic or conventional or traditional plant breeding, but these days we have something called marker-assisted selection. And this image depicts something that is done in big ag at a very extreme and mechanized level, where they're, instead of growing out the tomatoes to see which ones have the disease resistance and have the flavor genes, they're just taking a piece of the embryo or some part of the seed that has the DNA, and they're just testing that right then and there to see if they even want to grow out the seed in the first place. Now, for most purposes, especially if you're a gardener, you're probably not going to need this. Number one, it's incredibly expensive, and it's just, if you're a gardener, you might not even have access to the type of equipment and materials needed. They're essentially, you essentially don't need it. But I just wanted you to be aware of it. And as I mentioned, that this image depicted here, it's a very big ag, very precise way of doing it. But I consider marker assisted selection also what we kind of did with the tomatoes. You can also consider that, because a marker essentially is some region or some gene in the DNA that we know correlates to a certain trait. It may not be the gene itself. It typically could include a larger region. Like it's, for example, a marker is like, remember the warehouse reference? It's like saying, um, well, we don't know which page on which book it's in, but we know the gene we want. It's in that file cabinet, and it's in that specific drawer. Now we don't know which book it's in, but it's somewhere in that drawer. And that's really what a marker is. And so we could say that, oh, for example, like I did with roses, there's a variety called bright eyes. We know that it has some very dominant uh, black spot, which is a disease, a fungal disease, resistance genes. And so we can tell when evaluating its offspring which of those genes have been passed on because we know it'll either have it or it won't via trialing. And that is something I also consider marker assisted selection. Now, genetic engineering gets even more deep into uh, the weeds. It's a scuba dive. It's a deep sea dive into to plant breeding, and it really has kind of evolved along with plant breeding. It's uh, it's used a lot with initially with E. coli and bacterium, just because they're very easy to genetically engineer. They're the entire DNA of a bacteria, for example. Um, you'll very often find that it's just basically a big ring and call it a plasmid. And it's very easy to genetically engineer a bacteria. You just by they do a heat shock treatment or a gene gun. There's various things you can do. You have the gene that you want, and you just insert it in there, and it's very easily expressed. Now, that's a lot harder to do with something like a plant. But what we can do is something that's shown in this picture is we use agrobacterium. And it's a very unique organism. It essentially can colonize a plant's DNA and it'll form this weird undifferentiated mass, like a ball of just stuff. And what it's doing there, it's integrating itself into the plant's uh, genome, essentially, which is its DNA. And so by genetically engineering the agrobacterium, we can subsequently genetically engineer the plant. And that gets into a lot of other concerns that people have very strong opinions about. But there are many crops and plants today that we wouldn't have without genetic engineering. For example, the papaya. Uh, Papayas would have almost gone extinct due to this. It's like a ring spot virus, but they created the rainbow papaya. It's a genetically engineered papaya that's resistant to the virus. And if it weren't for that, the papaya industry would have been devastated. And even you talk about something essential as cotton. Um, Approximately 94 or 95% of all cotton grown around the entire world is genetically engineered. And that's for something like insect resistance and herbicide resistance. So when you're wearing your clothing and pillows and sleeping at night, just remember that you probably wouldn't have that if it weren't for genetic engineering. Now, people have different opinions when it comes to food that you can eat, but that's a video for another day. I just wanted to end by saying that, you know, this is a basic intro, and the basic thing I want you guys to remember is plant breeding, it's just customizing. We're just customizing and modifying new plants, creating new crops to be however we want them. If we want a yellow tomato, we'll make a yellow tomato. If we want a flavorful tomato, that has disease resistance, we'll do that. There's essentially an infinite number of things you can do. It all relies on your creativity um, and your dedication and time and space and efforts. But I really encourage you to try it out because you'll be surprised how easily you can do some of these things and how fun and rewarding it really is. So we've got lots to talk about next time in other presentations. But for now, I really just want you guys to go out, if you have a garden, go plant something and just do some cross pollination, start making some crosses, start saving seeds. Just have fun with it. Remember, it's all about having fun. So with that in mind, um, I'll end this video, and I hope you guys take care. And remember, don't forget to like and subscribe to the channel. It really helps out a lot, and I appreciate each and every one of you.